Hey guys, I am super excited about this chat with Dr. Melissa Oddie because as you guys know, we are still seeing huge colony losses in America on the commercial scale. We are seeing beekeepers that have lost up to 100% of their colonies, and we need absolutely all the help that we could possibly get. Whether that be from Trump or RFK, something needs to be done, and we need to figure out what is going on and how we can help our bees. So today, we are talking with Dr. Melissa Oddie about her newest research on viruses and Varroa mites and how that affects our bees. Come on. All right, so we should be rolling now. Hey guys, welcome back to the channel. Today I have Dr. Melissa Oddie back on the channel. If you remember last year, we did speak with her. She was finishing up one of her projects in Norway talking about some of the beekeepers out there that were actually finding Varroa resistance in their colonies, which was super cool. They were seeing like uncapping behavior and lower viral loads too. So if you haven't seen that, definitely go check that video out. Study out is all about viruses and how Varroa mites have affected that. It's called The Shift in Virus Composition in Honeybees Following Worldwide Invasion by the Parasitic Mite and Virus Vector Varroa Destructor. So there's a lot of fancy words there. Can you kind of explain like what the study was about and what you were finding?
Right. So basically, the data was taken a number of years ago when Veroa just started the spread. So we had a couple of areas that had Veroa, and then a couple of adjacent areas in the same countries that didn't have Veroa yet. So these were small islands, or these were just locations further north, or anyway, so they didn't have Veroa. So what the project basically did was took samples from these areas that had Veroa and the areas that didn't have Veroa, and they did kind of a full scale analysis on the viruses, about six or seven major viruses,
Mhm.
and there are over 20 of them. So we did a number of them. I'm not exactly sure how many, but so
Yep.
why we did that was to look at how the virome changed um, when the parasite was introduced, because we knew by that point that it was a vector for deformed wing virus. So we saw changes in deformed wing virus as we expected. But what we didn't expect was that it would affect other viruses so severely, even viruses that didn't necessarily use the mite as a vector. They were taking advantage of the system of the weaknesses introduced by the parasite. So we found changes not only in the prevalence of the viruses, so how frequently we found infections in the colonies, but also in the quantities of viral infections, so the number of copies of virus in the samples that we took. And all of this to say is that there are changes that occur when something drastic happens, like you have a parasite invasion of something that wasn't there before. It can affect the entire system. And that's what the paper demonstrated. It demonstrated that the viruses that might not even be associated directly with the parasite can change because the whole system has changed.
Yes, definitely. Yeah, that's I know viruses are it's starting to be a hot topic when it comes to beekeeping because we have so many different factors affecting our bees. I know right now here in America, we're seeing huge colony collapses right now, and there's so many ideas that are being thrown around with like what people think it might be, and viruses could definitely be a huge factor just because of the Varroa mite. So
It's possible.
for somebody who has never heard of like virology and why this is really a big problem for beekeeping, can you kind of explain it and like why it should matter to them?
Yeah, right. So we'll go to the kind of the simple saying that everything exists in a balance, like a
Yeah.
delicate balance. Um, and this kind of balance is dynamic in that things evolve and things shift, and sometimes viruses kind of evolve a trait that allows them to become more successful, and sometimes other viruses outcompete those. They work pretty much like species in an ecosystem, except they work
Mhm.
internally in honeybees rather than externally in an ecosystem. Um, So when you introduce something that changes that system, like you have Varroa, which is a vector for deformed wing virus specifically, but for viruses, what that does is it gives viruses that previously didn't have a direct pathway into the bees that direct pathway. So it means that they can spread much more efficiently, they can spread at much higher rates. And when you introduce that, you can basically open the door for a lot of other infections because they take advantage of the weaknesses. There are some viruses that get outcompeted, and all of this can change, how your bees interact with the environment. If they are kind of weaker from infections, they're less good at collecting food. They're less good at defending the hive. So, sick bees don't perform as well as healthy bees.
Hmm.
And these changes a lot of the time, especially for viruses, you can't see them until the infections get bad enough to start killing your bees. But all of these changes, all of this dynamism is going on, in your colonies day to day. Um, and yeah, so thinking about that and having scientists research that and kind of explain what could be going on gives beekeepers more insight into, the decisions that they might make for their colonies.
So in the paper, you guys were pretty much comparing colonies that have never seen varroa mites, correct?
Yeah.
And then colonies that have varroa mites abundant in that area. And what you guys saw is that varroa mites was in fact changing the type of viruses that you were seeing in the colonies. Was that correct?
The frequency and prevalence of some viruses changed. Like I said, that we
Okay.
tested a lot of different viruses. I think there were 6 or 7 that had kind of significant or interesting changes when
Okay.
Varroa was introduced. I mean, the first and foremost being deformed wing virus, which we knew was spread by Varroa.
Okay. Um, yeah. So if you could rewind, sorry, I was like trying to figure out where to go with that because you kind of answered the other question that I
Oh,
had.
oh, okay.
Um,
No worries. I'll just
no, yeah.
so I've got some
So
notes.
what the paper was kind of showing though was that varroa mites did in fact make a difference in kind of like what we're seeing with varroa today with or with our viruses today. So do you think if varroa mites never actually just imagine varroa mites never existed, do you think what we'd be seeing in our honeybees today would be a little bit different, or do you think it'd be the same?
Well, at that time point, it would probably be a lot better for the honeybees, because we wouldn't have had this vector that gave the viruses such a great pathway to infection. So the cool thing about deformed wing virus is it's not a new virus. It didn't come with Varroa. It was actually present in colonies pretty much all along. But
Um,
what happened was that it was the first virus, and it got really good at doing this, to use Varroa as a vector. So in the Varroa basically takes hemolymph and fat body tissue from the bee. The virus particles get stuck to the proboscis of the mite, and then when it transfers, the virus stayed alive long enough to actually get put into another bee. Humans have pretty serious vectors as well. So, the mosquito transmits a lot of different diseases, and some diseases can only be transmitted via the mosquito as a vector. So, deformed wing virus basically adapted to use the mite as a vector, and actually only about 1 to 2 strains at the beginning of the global spread could actually do this. And there's a really interesting paper by Stephen Martin when he was looking at Varroa introduction in Hawaii. That we had kind of a time lapse of Varroa introduction based on the islands because they were introduced progressively by island. And when he did a viral analysis, he found that actually the strains of deformed wing virus were quite diverse when Varroa wasn't present. But as soon as Varroa was introduced, you had a massive loss of viral diversity. So there was only one strain by the end of the study, when you had Varroa introduced for 4 years or more. One strain that was kind of showing up all over the place, and the other strains were just not present.
maybe there's just some other factors that Varroa, since Varroa is making like the colonies a little bit weaker, that's just what that virus maybe thrives in,
It
I'm
was
guessing, for why
not
it's
all the
so up.
strains learned to use or adapted to use Varroa as a vector. So the strains that didn't do that died
Ah,
out because they were out competed. So it's not to say that
okay.
patterns like this would never happen if Varroa hadn't come, because everything is evolving, everything is shifting. But the introduction
Mhm.
of a vector is usually something that creates really big changes. Mostly for the worst for the hosts that have to deal with it.
Okay. So, okay, this question's probably going to dig really deep into virology, because this study kind of looked at virology as a whole and how it's affecting the varroa mites and the honeybees, of course, but it was focusing on like the virus composition shifts. Why is that more important than simply just looking at like the virus prevalence? Can you kind of explain
Oh yeah,
like
cool.
what
So
that would
there
mean
are
then for
two.
somebody who knows absolutely nothing?
Yeah, so there are kind of two sliders for if you can visualize that for the kind of the importance of virus infections. There's the viral prevalence, which is if you have a number of samples, how many of those samples test positive for the virus. So that gives you kind of a relative frequency. So how many samples, how many organisms are infected, and then you have the quantity of viral particles, and that's the second. So that's the severity of the infection. So that's how bad the infection
Okay.
can get. So if we're going to take deformed wing virus as an example, the prevalence and the abundance were affected by Varroa. So that means the we had more cases of deformed wing virus. So more hives were testing positive for deformed wing virus in the presence of Varroa, and the viral titers, so the level of infection was much higher. You have something like black queen cell virus, which actually was affected by Varroa. So we had an increase in prevalence, as I recall. So we had an increase in the number of cases, but the intensity of the infection did not change drastically with the introduction of Varroa. And we know that black queen cell virus can be transmitted by Varroa, but not to the severity that deformed wing virus can be spread. So that's probably why we didn't see like a difference in the titer, in the intensity of infection. If we look at Kashmir bee virus, which apparently it didn't really have an increase in abundance. So the number of cases of Kashmir bee virus didn't increase with Varroa, but the intensity of the infection did seem to have, like Veroa did have an impact on that. So what that tells us is that maybe Kashmir bee virus isn't, can't use Varroa as a vector, but it can certainly take advantage of the weaknesses introduced by deformed wing virus and by Varroa to increase its infection level. So that's why prevalence and viral infection and quantity, that's why they're important, because it tells you not just how many cases there are, but how severe the infection is going to be, and how that impacts your bees.
Definitely, because to my understanding, viruses are always in our honeybee colonies. It's just if the load
Always.
gets really high, that's when we start seeing negative consequences.
Yeah, yeah. And the more
Okay.
challenges that the hives have to deal
So
with. Oh, sorry. We had a lag there, I think. The more
yeah,
challenges
no,
that
go ahead.
honeybees have to deal
Yeah.
with, the easier it is for these infections to take hold and actually cause damage.
Yeah, definitely. I've been learning a lot lately about pollen and just how important it is for our bees, especially in the late fall time. And I honestly wonder if some of like the varroa mites and the viruses we're seeing, it's kind of hinging on the fact that our forage has been not really the best over the last couple years, just with like the weather and some other competing factors too. But um,
Yeah.
yeah, I definitely think that viruses are going to really be capitalizing on that if the bees aren't healthy enough to be able to fight them themselves.
So that is absolutely a component, is the environment that
Mhm.
the bees are raised in. And we know that nutrition is so crucial to honeybee health, and that, if they don't have good nutrition, they're more susceptible to the effects of pesticides, to parasites, to diseases, and stuff. It's just like in humans. If you have a poor diet, if you don't eat well, then you often get sick more often.
Oh yeah, definitely. So you did mention that deformed wing virus was kind of one of the ones that you saw that was benefiting off of Varroa. Was there anything else that you saw too that was benefiting off of them the most?
Yeah, and that's interesting. So we know that some viruses can definitely use Varroa as a vector to kind of hop from bee to bee, and
Yep.
that's really important because it having a vector basically bypasses the first line of honeybee defense. You get past the exterior, which is supposed to
Mm-hmm.
keep
Yep.
viruses out. You just kind of get a direct pathway to that. Um, so you have that, and then you also have the viruses that can take advantage of weaknesses introduced by higher levels of virus. So we know sac brood virus, the SBV there, and black queen cell virus can be transmitted. There may have been one more, so it can be transmitted by Varroa. But then we have things like the Kashmir bee virus that might be muscling in and taking advantage of that, and other viruses as well. I'm going to ask you to repeat the
What
question. Did I get
exactly is the
everything?
cashmere? Yeah, no, you did.
Yeah.
What is the cashmere bee virus? I honestly don't know anything about it.
Yeah, not many people do because it doesn't normally provide like very severe infections. It's one of the paralysis viruses, and they
Oh,
not a virologist, so I'm gonna tell you what I know. Um, Basically, these are viruses that can become very severe infections if the hive is already weakened. I think in most cases, the paralysis viruses, some of them can get pretty intense, and what you see is bees that are moving very slowly or moving kind of off at the entrance. Under the exact conditions, I'm not sure when that the infections would take over and actually become a problem, but because deformed wing virus and Varroa was such a much bigger problem. That actually has the potential to kill your hives very quickly. These paralysis viruses
mhm.
generally don't cause a problem by themselves. So
Oh,
yeah, so KBV, IABPV, those they're present in colonies. They're present in a lot of colonies, um, but generally they're considered lower threat than something like deformed wing virus with Varroa in the mix as well.
okay. That's good to know at least. Hmm. So you guys also, I saw in the study, you were pulling bees from different areas around the world. So did you
Yeah.
see any differences just in like the regional area too?
Yeah, so in this particular paper, I was responsible for doing the initial statistical analyses, and yeah, so area was definitely a very significant factor, and that is because the environments are completely different where we were pulling these bees from. The methods of care were different. The environment has a very huge impact on honeybee health and how infections spread and how mites spread. So yeah, there was kind of a large impact of area. It's very difficult to tease that apart from other things, though, because there is so much variation. Um, so you have
Mm-hmm.
even between apiaries or between beekeepers, you can sometimes have differences that just completely change things. So we found an effect of country, but I would say I would add that in, and that beekeepers probably an effect, and whether it's on top of the apiary is on top of a hill or at the bottom of a hill. You can have changes within 1 kilometer, even though the environment is
Oh
technically the
wow.
same. You can have microclimates that change. Um, so yeah, so it's very difficult to pick up on all of these things that affect. So this was a massive study. How you mitigate some of these effects in a study is you basically just take a lot of data. So we took a lot of colonies. We analyzed a lot of bees, and tried to capture kind of the largest trends in the data, and that's what we came up with.
I heard that in tropical climates that Varroa doesn't thrive as much because it's humid. Did you guys study any hotter, more humid climates, or are they all just kind of like northern?
This was northern. specifically because I think we were dealing with European honeybees, and we wanted to basically keep the pure stock European honeybees for the study. If you go
Makes
further
sense.
south in North America and also in Africa, you get Africanized bees and African honeybees, and they are actually much better at dealing with Varroa. So that would introduce a bias to the data that would probably
Okay.
make it harder to see patterns and stuff. But generally, Varroa originally came from a very tropical climate. It came from Asia.
Mhm.
So they generally actually do better
Oh.
in warmer climates and in hot and but it has to be humid as well. So hot and dry doesn't work, and cold and wet doesn't work. They like hot and wet sort of climates. And I think
Oh.
it might be just
Okay.
because the survivability outside a hive might be higher.
Okay, that makes sense then. I have to remember where I heard that.
They don't do very well in Norway, so
So
I know that.
really? Yeah, I know the brood break seems to really help with keeping the Varroa mites back that we get in the winter time, so that
Definitely,
would
yeah.
make more sense then. So if you were to design a follow-up study after this to kind of dig into any unanswered questions that you have, like where do you want to go from this after this study? What's on your mind? What are you thinking about?
Well, the study taught us that basically if we see something like a drastic change, like the introduction of Varroa, we should start looking at other elements of the whole system that could be affected. Um, so this is actually something like this has happened before in a case called the Isle of Wight disease, and a lot of beekeepers have probably heard of it. It was over 100 years ago when you just had a mass die off in the UK and nobody really knew how or why it happened. Um, Now we know that it was likely something to do with tracheal mites and possibly a virus that was introduced, just like Varroa. And that was almost the end of the British black bee. And from there, you had brother Adam and the invention of the buckfast bees, which was bred initially from the remaining black bee stock and Italians. Um, so if we had the technology we have now back then, we might have looked for the virus. We might have been able to actually find exactly what happened and why all the bees died. But now that we have this technology, we can actually start looking at that. So if we have mass die-offs again, we're like, okay, well let's run a viral scan. Let's see if there's anything that changed in the virome. Let's see if there's a new parasite that came into
Mhm.
play. So if I was going to design a follow-up study, I think I would start focusing on kind of the quantity and the genetics of the viruses and how they're changing, because they are always changing. And it might be able to give us an edge to predict maybe the next step in this dynamic pathway and what to watch out for.
Definitely. I'm gonna kind of have my tinfoil hat on a little bit, and it kind of feels like sometimes because honeybees grow really fast, and I almost feel like if there's not something to kind of hold them back, we would almost have too many honeybees. So it kind of feels like there's always like a new mite or a new virus or something kind of like holding them back. It's just trying to find that balance. That's the hard part.
Yeah, I mean, there's definitely a
Okay,
lot that affects
so
honeybees. I'm trying to work off this
um
lag here. There's definitely
yeah, no, it's
a
okay. Me
disconnect.
too. It makes it a little bit more difficult. I got to get used to talking to people online. It definitely changes things a little bit.
There is a giant snowstorm outside, so I'm gonna blame it on that, the dodgy connection.
Yeah. Oh, definitely. Yep. So some might say that focusing on viruses can kind of be a distraction from some of the other things that are going on with honeybees. I am one of those people that have said that before, saying that we're focusing too much on varroa mites. But how do you like? What do you think about that?
Oh boy, so you've touched on kind of one of the biggest questions is in academia is where do we put our resources? Because
Hmm.
it's never a simple question because you have all this interconnectedness. Um, so what is too much attention? What is not enough attention? there are so many unknown variables, like we're learning, like with this study. It's, well, actually, Varroa affects not just one virus, but a whole bunch of viruses. And how does that impact the
Hmm.
system? So if you're looking kind of at a broad spectrum, yeah, pesticides are really important. The fact that we're losing good forage, very important. Those things would be drivers to make things a lot harder for beekeepers. But then you have things
Hmm.
that all of a sudden, all the bees in this area are dead. Why? What happened? And things like viruses could be playing a role. And if we don't study them, it'll remain a black box. But
Hmm.
as we put research into this, as we learn the patterns, we can apply them to cases outside of the one that we're studying. So if we have a good grasp of how viruses work and how they react to things like vector introduction, or things like, higher pesticide risk, or a lower quality forage, we can make predictions as to how these things would change based on, the changes in their environment. So what's more important? It's so hard to say, because if we have this background knowledge, we're better prepared for things that can happen in the future. So I think a little bit of everything is definitely a positive thing. um, Yeah, I wish I had a better answer than that, but so we just don't know what's important in a lot of these systems.
Yep, exactly. And I think too, this the more that we study it, we're kind of seeing, like you keep talking about, that it's all a system, that everything works together and it's all connected. So if one thing starts to fall out, then the whole system collapses.
Yeah,
So really, yeah, it's everything, everything that needs to be
it is
looked at.
everything, and it's so hard to make decisions on what's more important than other things.
So can I ask for your opinion on what's going on currently in America right now with all the colony collapses that we're seeing? Have you looked up on that a little bit?
I haven't, actually. So why don't you give me a bit of context, and for
Okay.
the viewers as well, as to what exactly is happening and where, and I'll do my best to give you an opinion.
Yeah, so yeah, definitely. And I know all we can really do is speculate because they're trying to study the bees and figure out what exactly happened. But it's mostly from what I'm hearing in the commercial industry. I've talked to some hobby and sideliners, and personally in my own operation, we're looking just fine. But we're seeing huge die-offs where the the beekeepers were pulling bees out of their sheds or just pulling them to send them off to almonds this year for California, and we weren't able to really make that quota just because a lot of these colonies are pulling out, were collapsed, and they're seeing a higher percentage loss than normal.
Okay, so the information we have is basically the hives that were slated for almonds. A lot of them just didn't make it through their overwintering.
Yeah,
It's
yeah.
very difficult to speak to that if we don't have any sort of data. So if I found a, die-offs, I would start taking Varroa samples first off and see if the Varroa got out of
Hmm.
hand in different colonies and stuff. Then I would probably screen the bees for the viruses. Probably check for
Hmm.
other infections, possibly. If you have winter die-offs, it can rule out some things, than other. Like, we're not really sure what condition the hives were in. Kind of their characteristics for fungal infections like nosema. I think any of the foul broods, the beekeepers would probably know that they had those before anything got
Hmm.
drastically bad, because the symptoms are quite easy to detect. Um, it is possible that, one of the viruses actually has made an adaptive shift, and that it's just spread and causing more problems. But without more information, it's really hard to tell.
Hmm.
We don't know if there was a change in the crop spraying in these areas, so we don't know if there was a new chemical introduced. Um, we don't know about the climates. What sort of uh, summer and autumn they had before they went into winter. So, did they have enough food, or what was in the food? What type of food was it? These are all things that could affect seeing mass die. So, was it in like a large area? Was it like across multiple states? I'm gonna
It
look this
sounds
up
like it's
after.
just
Yeah, definitely. It sounds like it's just coming from all over the United States, not really in one
Okay.
particular area, so that's why it's so puzzling.
There's probably one other thing I could say to that. So I'm not sure if it's changed, but from what I know about the system, especially the commercial pollinator system, is that a lot of beekeepers get all of their queens from about 4 different breeders. And we can talk a little bit about how genetic diversity is a very important thing, especially for honeybees. And that if you shrink that diversity enough, you can actually facilitate things like mass die-offs, because if you have a whole bunch of queens that are genetically very similar, they're going to have the same weaknesses as each other. They're going to have the same strengths. So if you have one
Hmm.
particular strain of honeybees that is very susceptible to a specific virus or specific type of infection, or just not as good as at getting resources or something, then all of the others will probably have the same trait. So one of the things I harp on in my research and when I talk to beekeepers is you have to keep the genetic diversity high. And honeybee biology is actually designed to maximize genetic diversity. You have a queen that mates with multiple drones. You have genetic crossover rates are much higher than any other kind of eukaryotic organism. So most other animals. And what I mean by genetic crossover is that when you have cell division, when the chromosomes line up, when they're producing reproductive cells, a lot of the chromosomes basically share genetic information. They just trade back and forth. And with honeybees, that rate is much higher than any other animal. So they're very designed to maximize their genetic diversity, and this is because they don't have a very large dispersal rate. But when a colony swarms in nature, it doesn't go more than 4 or 5 kilometers away from the mother hive. So that means all the hives around each other are going to be very genetically similar. So it serves the bees really well to have a lot of systems in place where they're maximizing diversity. So yeah, so we have an animal that is designed to maximize its diversity, and then we're having basically, hundreds of thousands of hives that we get from a few queen breeders that are all very genetically similar. That kind of sets up for kind of large scale problems to happen. So I would say genetic diversity might actually be a player in what's happening now as well.
Yeah, that's actually something my partner and I have talked about because just with some of the queens we've tried in our yards, because he does a lot of queen breeding, and we've seen there's some bees that are resistant to like everything, but then there's other bees that are susceptible to sac brood or chalk brood and stuff like that. So yeah,
Yeah.
if you don't have that spread out of just different genetics in your yard, if one thing comes, then you're taking out everything.
Yeah, and I know it's difficult because it makes bees unpredictable. And we like predictability.
Yes.
We like to be able to tell what our bees are going to do. But it does having genetic similarity in all of your bees does introduce weaknesses.
Could that make it really difficult for breeding for Varroa resistance at all? Just because it sounds
Um,
like since they're constantly evolving and bringing in new genetics.
Interestingly, actually, high genetic diversity I think may help develop resistances, because what you're doing is you're increasing the probability that you have these surviving traits, and then if you select for these traits, you're kind of filtering your population and keeping all of the surviving traits, while other traits kind of get weeded out. So when the population I started with, from Tarja Reinertsen as a buckfast population in Norway. Basically, he was taking everything he could find and building his stock from that. And then what he did was he stopped treating completely, and he lost a lot of bees, but not all of them. And he bred from the survivors, and he continually took new stock in. And so the most of the bees now are exceptionally good. You get one colony or two colonies that are either too aggressive and need to be requeened, or they're not the best survivors and stuff, but that's a very low percentage of the stock. And I think having that equilibrium, so having most of your stock good, I think is probably a point where we can say, that'll do, and just keeping the genetic diversity high and taking some of the colonies that might have to be requeened or just not as good at performing as the others. So always have a small percentage of your stock that you're not happy with, but keep the genetic diversity high. And I think that's a way to keep a good balance.
That makes sense. And that's actually perfect because you are writing a book about that right now, about developing Varroa resistance in your queens.
Yeah, yeah. So I have partnered with two other very prominent researchers, Professor Marla Spivak and Professor Stephen Martin from the UK. We're teaming up to kind of plot all of our research, kind of write it out in these case studies, and also in not really an instructional guide, but basically kind of a step-by-step on how beekeepers have gotten resistant bees in the past. So yeah, and that's actually, we're going to be running a Kickstarter to fund that book. It'll be starting March 10th, so save the date. And we're going to be raising funds to try and produce this book kind of, quickly and efficiently. And we got a lot of really cool rewards and stuff for this Kickstarter when it launches. We've got some art, and you'll be able to buy an advanced copy of the book, but we've got some really cool poster art that is done by myself. And we've got this cute little teddy bear, which I'll showcase here. This is Melly, the honeybee, also designed by me. And so she will be a reward as
That's
well.
cute.
And we've been getting a lot of call for T-shirts. So there's if you go to the website, which is going to be on the slide and probably linked below, you'll be able to see all of these rewards, and you'll be able to read more about the book, why we're writing the book. You'll be able to read about the authors, myself and Marla and Stephen. And then you can sign up to our mailing list, and you'll get a notification when the Kickstarter starts. And for the first little bit, there's actually going to be a discount. So members that sign up will get linked directly, and they'll be able to take advantage of the discount when they back the project. So that is it. Good. Do you have any more questions? Do we want to go back to the virus thing a little
Super
bit?
exciting.
It's the lag. It's the lag.
Well, one thing just kind of hinging still. Yeah, you're okay. One thing still kind of hinging off of your book. So, one of the biggest things I've heard, at least in America, when it comes to, because genetics of our bees, I think, is a really hot topic just because of Varroa mite and trying to find Varroa resistance. And there's a lot of commercial beekeepers, pretty much the whole commercial beekeeping industry, because of the way we manage our colonies. Being able to manage like a treatment-free system is very, very difficult. So
Yes.
for these bees that you did see in Norway, one of the questions I've had from some other commercial beekeepers is, if we were to transition to a Varroa resistant bee, their main problem is having those bigger colonies later in the year. Because, I mean, for almonds, that is the biggest thing for American beekeeping, is they have to hit their quota for that, and that really kind of kickstarts the rest of the year for them. So having those 6 to 8 frame averages in the dead of winter in February when it starts is a really difficult thing. And I know some genetics, they like to have like maintain those smaller cluster sizes. So, I mean, just what have you seen yourself? Is that something that can be bred for to still have Varroa resistance and have those huge colonies? Or like how is it all kind of working together? Because I know the brood breaks of the winter also help with Varroa. I don't know, just kind of like throwing like all my thoughts out there with it.
Yeah. So there is good news, and that's why I want these co-authors on the book, is because there are actually examples in the States of Varroa resistant bees that are used for commercial purposes. I'm not sure if you've heard of the Pauline or the Pauline 2. They are bees that are bred for kind of not complete resistance yet, I think, but they are getting very, very close to doing that. And it's basically kind of a healthier bee. And what commercial beekeepers have done in working with this stock is they've taken a small amount of their stock, so 200 odd hives. They've done the selection on that, so they don't take a loss with their entire stock, but they're actually getting, genetics that might actually give the bees an advantage against Varroa, and slowly introducing them into their central stock. So that's definitely one way of doing it. Instead of throwing all your eggs in one basket and experimenting with your whole all of your yards, is you take a small test yard. It's very time consuming. You do have to count mites and treat and keep a closer eye on them. But if the result is you can slowly introduce bees that will give you an edge against Varroa, it might, serve bees well in the long run, because if you're taking care of the vector, you're taking care of the viral infections by default, and you're actually reducing the number of risks that your honeybees have to deal with, when they're in critical periods, like they're overwintering and stuff. So the answer is yes. I mean, Tadia for Norway had a very competitive commercial stock. It was they were honey producers, but they overwintered large, and they were very gentle. So you can absolutely have desirable traits in with your Varroa resistance. We've proven that. There's another paper actually about bees in Cuba, because when Varroa hit Cuba, they couldn't afford to treat. So the government basically just said, we're not going to treat, and we're just going to deal with it. So now we have actually very high resistances in Cuba. And they did a genetic study on these bees, and there is very little Africanized DNA in these bees. They're mostly completely European stock. So they have very high producers as well in almost the entirety of Cuba that are resistant to Varroa. So Stephen Martin wrote that paper as well. I can probably find the link for you.
Okay. Yep. So sorry about that. Gotta love the weather and bad internet connection. That's what happens when you do this stuff online.
Technology.
Yeah, definitely. Okay.
Yeah,
So
sorry.
where did we leave off? We were talking about Varroa resistance in queens. So we can kind of go back a little bit to your paper just to kind of close that part up. So there was a lot of cool findings in it. And possibly maybe we could find, do you think there's anything we could find from the paper that could like potentially lead to more studies on Varroa and viruses, or even just a product that might be able to help them with viruses? Where do you think the study could kind of go?
Well, in order to find a specific product, I think the pathway is going to be kind of like a little bit long, because each of these viruses is unique, and not all treatments or vaccines will work for all of them. But if we're talking about takeaways from this work and stuff, basically we're thinking the way we think about things as a kind of as a whole system is definitely something that we should be working towards. And in terms of future research, the really interesting thing is that we have tools that are just getting better and better, and we're able to analyze this data, so make viral profiles or predict what the viruses are going to do much faster. And interesting, like machine learning is actually helping us in leaps and bounds, quantifying this. So we have these artificial intelligence algorithms that are able to analyze mass amounts of data, like genetic data, like medical imaging data, and they're able to find patterns that humans haven't found yet, simply because they can analyze just massive amounts of data, very quickly. So these tools are probably going to become a lot more prominent. And what we can use them for is we can use them to kind of advance drug trials and make these predictions, so that we can speed up the development of treatments, or we can use them to get better predictions on how the virome is going to change if we introduce certain elements and stuff. So that's really just kind of the frontier. We've just scratched the surface of what this technology can do, and we're still learning. So although AI has been used to, or a lot of the stuff it's been used for maybe isn't that popular, but for medicine, it's actually going to become an invaluable tool. It is very helpful.
Definitely. That's really cool that we're actually able to start doing that. And I kind of wonder if that kind of helped with like the vaccine that we're now seeing that they're saying is helping with deformed wing virus, but they originally found it for American foul brood. Have you looked into that at all? I don't personally know a whole lot about the vaccine. I haven't tried it myself, but I don't know if that's
The
maybe a
last time
route.
I checked is they were still running trials on it, but it seemed very promising.
Oh, okay.
Is this
Okay.
the RNA strand vaccine?
I
I'm not
believe so,
sure which
yeah,
I'm not sure
for
which one it is. Yeah. So I mean it seems like it works. The problem is it is a vaccine. I think it needs to happen. You need to actually boost your hives with it on the regular. And any hive that isn't
uh-oh.
vaccinated, you have to get it vaccinated. And then, so the question is, how much does this cost? Is it going to be less expensive than treating your hives with other means that we already have? A lot of methods are ingenious and they're great, but they end up being too expensive for the average beekeeper to
Mhm.
use. So
Yeah.
we're still working on it. And the translation of all this cool science into a practical thing that is also cost effective, we're still working on that transition.
Yep, definitely. Well, I guess that's most of the questions I had for the paper. I'm really excited about it because I think we've always kind of noticed that Varroa seemed to make the viruses really go up, but it's nice to actually see some data behind it of what is actually happening in our
Yeah,
hives.
yeah. No, so it's definitely cool. So yeah, keep in mind that whenever you have something affecting your system, there's a lot of other elements that you might not see that are also being
Mhm.
affected.
Yep,
So yeah,
definitely.
and good luck out there. And for all of those experiencing high losses right now, I'm very sorry. Hang in there,
Mhm.
because we're getting better at doing this, and we'll be able to find solutions faster and faster.
Oh, definitely. If I've learned anything, it's that beekeepers are just as resilient as honeybees, so we'll find a way.
Exactly. All right. Well,
And
thank you
definitely
so
go
much for
check
having me
out.
on the show.
Oh,
Sorry.
yeah, definitely, no problem at all, and I'm super excited about the book you're writing. So, you're starting you're gonna start writing it in March, and then it should be ready, I think you said the end of the year?
Well, we're gonna try and do that. If the
Okay,
Kickstarter goes through and I can actually devote 100% of my time to writing the book, then it'll happen a lot faster.
okay.
But even then, if the Kickstarter doesn't end up working out, I still want to put the book through. It'll just take a lot more time. So the fundraising
Mhm.
project is really just to make sure that this gets done. You know, kind of now when people are talking about it, when they need the information, and there's a
Yes,
lot of really
yep.
good information that we have to share, that I think would just, the book format would be just the best way to get it across. And
Mhm,
to make sure all of this scientific data gets crunched into a format that beekeepers can read and access easily.
mhm. Yes, definitely. I think it's needed, especially right now with everything that's been going on. There's a lot of changes that need to be made to the beekeeping industry in America, and I think it could definitely go hand in hand with helping some of that.
Yeah, it's all about keeping that balance.
Yep, exactly. All right. Well, I guess that is that. Thanks for watching. And as always, don't quit and be fit. And honey is just a product until you meet the beekeeper behind it. I'll see you guys in the next one.